So the first step of propagating genetic information within an individual cell is transcription. Now it's very important to understand that we have a certain level of organization within our genetic material, and we've talked about this in the previous chapter. DNA is carried on chromosomes, but what we've tried to introduce in 12.1, and we'll introduce it again, is that those DNA sequences are organized into discrete units called genes. So think of a gene as a functional unit. It's that gene that carries the information necessary to produce products like proteins. For humans, when you look at all of our DNA, of which there are many, many bases, 2% of our DNA is organized into genes. Think about that. It's like having a book and only 2% of the pages in your book actually have anything of substance. The others doesn't necessarily encode for a product, doesn't necessarily contain information. I mean, that's staggering, right? So for an organism to express their genes, they have to find them first. And then once they find them, they have to transcribe them. So how can we do that? Well, here's a basic overview. And while it's not labeled, I'll try to do so now. Everything in pink here represents a gene. That has information that ultimately is going to drive the production of mRNA. It's going to contain the code in order to make this mRNA molecule. We also have other regions that we're going to highlight in this chapter. This is a regulatory region. Remember I just told you that 2% of our DNA is contained within genes? Well, how are we going to find where a gene starts? Well, these regulatory regions are how we identify the beginning of a gene. We also have a terminator. How are we going to know where the gene ends? Well, we have a series of specific sequences that result in transcription being halted. And so what we have is an mRNA that corresponds to this region, but we also have accessory sequences like regulatory sequences and terminator sequences that help to identify the start of a gene and help identify the end of that gene. There are 3 stages in transcription. You should be able to describe what's happening at each stage: initiation, elongation, termination. Initiation, this is where we have to recognize the gene. Now, in order to do that, in bacteria, there's a protein called a sigma factor that allows RNA polymerase to recognize a region called a promoter region. I'll show you where that promoter region here is in a second. This stage is completed once the area in and around the promoter is melted, exposing the templates and forming an open complex. That's the committed step. Once you reach that step, we have to copy the DNA. So let's look at this. I know this is busy, but we'll try to break it up into parts. In blue here is a promoter region. The promoter is part of the regulatory region that identifies the 5' end of a gene. See, there's a protein that does all this, right? It's a polymerase. Just like DNA polymerase made polymers of DNA, RNA polymerase makes polymers of RNA. And so how does this enzyme know where to begin? Well, it's going to bind this promoter region through one of its subunits called a sigma factor. Once it's docked at the appropriate promoter, at the appropriate location, the enzyme initiates the opening of the DNA template, the separation of the strands. No helicase required. RNA polymerase has the inherent ability to separate the strands and break through those hydrogen bonds, forming what we call an open complex. That is the committed step. Once the DNA strands are separated, we're off to the races. So now, what do we mean by off to the races? Well, that's the second step, elongation. That RNA polymerase is going to read the template strand in order to synthesize RNA from a 5' to 3' direction. So check this out. Here's my DNA. Here's one strand, here's the other. Here's a promoter up here. Because a promoter is here on the left-hand side, RNA polymerase binds, and now this is automatically going to become the template. Because we read DNA 3' to 5'. We build nucleic acids, both RNA and DNA, 5' to 3'. So now, let me choose a new color. So now here's where I'm going to start with my RNA, and I'm going to build it in this direction. So this top strand is a template strand. The bottom DNA strand is what's called a coding strand. Now let's think about why that is. Well, here we go. If this is an ATT GGC, let's say that's a series of nucleotides. We know the opposite strand of DNA is going to be TAACCG. All right. So now, let's bring in another color. When I use this as my template, I'm going to build the RNA, right? So if there's a T here in the RNA, an A will be placed. If there's an A in the DNA, a U becomes the base pair. A U becomes a base pair. G, G, C. Notice now the newly synthesized mRNA is complementary to the template, but it's almost identical to the coding strand. The only difference is that RNA uses a uracil instead of a thymine as one of its building blocks. Other than that, A is at the appropriate position, G, G, C. The U's are exactly where the T's would have been. That's what makes this the coding strand and the opposite DNA strand the template strand. So here we go. So now DNA is moving 3' to 5' down the template, building 5 to 3. So it's elongating and growing from 5 to 3'. So this is going to get longer and emerge more and more out of RNA polymerase. Here we can see the active site, what's happening. So now I'm building in this direction. So if there's a G, a C is going to get put in, a G, another C gets put in, growing 5 to 3. So we're going to continue to do that throughout the entire coding region until we reach a series of DNA sequences called the Terminator sequences. At this sequence, the DNA polymerase is going to fall off the DNA, and it's going to release the transcript. So the region which gets copied in the process of transcription is the region from the start site all the way to the terminator site.